strand. So if you have one strand, you have to have the second strand, and then you have other things too. That's what we're going to get into today. I just wanted to show you how many base codons that you have. How many base nucleotides? Each one of these letters is a nucleotide. Okay, it has a sugar, it has ATP, and it has a phosphate, and then it also has a nucleotide. Okay, one of the base sequences. Okay, so here's all the base sequences. Now, why is a protein important? Okay, this kind of gets us back to why a protein is important. Yeah, proteins are formed from amino acids. That's what's so important from it. So proteins come up to us by amino acids. Amino acids are created during the transcription, or tRNA, when it meets mRNA in the ribosomes. Remember the ribosomes? The ribosome itself is made up of two components, an upper component and a lower component. And inside that component is the RNA, the rRNA, the ribosomal RNA. And that ribosomal RNA has what is that enzyme that's inside of it that glues it back together? Do you remember?
Polymerase.
Polymerase, good. Polymerase. Now DNA gets glued back together inside the nucleus by polymerase 3. But in RNA, it's just called polymerase. Okay, so it's an enzyme that does that. That puts those together and then forms a 3-base sequence or anticodon that has an amino acid attached to it. And that amino acid, when you get a whole bunch of them hooked together, okay, then they form what's called a protein. And a protein can come in the form of meat. So we have protein in the form of meat. What does our body do? Our body takes the meat and breaks it down into these amino acids that are found, that are broken down in our gut when we dissolve the food. Then different parts of our body takes these amino acids and uses them. And then finally, once it gets through the body or through the blood, it transfers into our muscles. And so now we can say, if you eat meat, that will grow muscle. Meat turns into muscle, which makes sense. If you think about God's creation, it makes perfectly sense. We're eating muscle. We don't think of it that way. We're eating ground beef, but that is the muscle part of the animal. We break it down with our body, and it turns into to be fuel for the muscles of our body. Okay? All right? Then also we have some other things with protein here. This is called collagen. Does anybody know what collagen is for? Collagen, maybe your parents take it. Okay? Cramp? Okay. All right. Well, I don't know. She was grabbing her leg, and I know a lot of people said that they were cramped after the activity today.
I didn't feel anything.
Anyway, collagen. Collagen is the stuff that works inside of the joints. So, for instance, people that have arthritis, okay, sometimes they'll take collagen before they get there, or they have hips that have trouble. They'll take collagen because collagen is actual protein that ends up being utilized there, so we don't get the effects of those proteins being overused. Okay, so in the proteins, the enzyme regulates those chemical reactions I have here, and then the body uses it to build, okay, and also to operate different parts of the cells. Okay, so that's really what proteins do. Now, we're not here. Okay, we've already talked about tRNA then, if we're here. We did do tRNA? Oh, okay.
Went over it.
I'm so
Yeah,
confused.
okay. That's helpful. Because I don't want to go into mutations here today yet. Okay? But, so tRNA, let's go ahead and get the lights on then, and we'll talk about mRNA, tRNA, and the other things. Okay? And this is what's important. This is what's important. This is the culmination of everything that we've been talking about. So if you have those worksheets that have all those letters down the second column, that worksheet, I'm going to give you an example of what we have here, and then we'll take a sample, and so you'll know what you'll end up having to do. Okay? What I have up here on the board right now is what? Tell me what is on the board right now. You should know.
3-base sequence.
Okay? No, we do not have codons.
A 3-base sequence.
I separated them this way only to put them into codons, but really it wouldn't be this way. They're base sequences, but you got to tell me more. What is this? You should be able to recognize right now what this is. Anyone? It's not an amino acid. It's not a protein. It is a DNA sequence. In fact, it's one strand of a DNA. So what has happened here? Very good. It is unwound and unzipped. So it is unwound and unzipped, and what unzips it?
The thingy.
Heliocase.
Heliocase. Good. ASE means it's an enzyme, so the enzyme heliocase has separated this, and it gave me one strand, usually called the alpha or the beta. Most biology books list it as A and B strands. So we have one strand, one side of the DNA molecule. That's all we have. So we have the side, and we have the base sequence, okay? So now, you have to be able to do several things, okay? So help me out here, okay? First thing I want to do is this strand unzipped and unwound, or unwound and unzipped for what reason?
Duplicate.
We don't call it duplicate.
Replicate.
Replicate. It wants to replicate. So it's replication. What carries on the replication? What does the replication think? What carries on the replication? mRNA.
Really?
Very good.
Dude, good job,
So
Royce.
it's mRNA. So mRNA is going to hook into the DNA. Now, are we there? Do you understand what's happening here? So it unzipped, and this DNA strand is floating inside the nucleus. It can't leave the nucleus, okay? But it unzipped because it wants to do one of two things. Either it's going to make a copy of itself and zip back, or it's going to send a message outside of the nucleus called the mRNA. That's replication. Duplication is when it makes a second copy of the DNA. Okay? So now, mRNA. mRNA says that I'm going to hook in the base sequences with each other. So what base sequences go together?
GC.
C and G.
Yeah.
So cytosine hooks up with guanine. So wherever you see a cytosine, let's put a G. There's a G, G, G. Those are all the G's. Now what about the G's? What do they hook up with?
Cytosine.
Cytosine. So wherever you see a G, that's going to hook up to a cytosine.
Okay.
Cytosine, cytosine, cytosine,
So,
and that's
all.
it. Okay? Now we get to the last ones, adenine and
Thymine.
thymine. So kind of tell me what we got to be careful of.
The
Uracil.
yeah.
What is uracil?
The RNA. It's
RNA form of thymine.
yeah.
That's an excellent way to say it. It's the RNA component of thymine. So wherever you're supposed to have thymine, you must substitute a U. So what is thymine going to hook up with? Adenine. So there's an adenine. It should hook up with thymine, but no, because we're in RNA, this is going to be uracil. Uracil, uracil.
So then do we put an A where all the T's are?
Awesome! Awesome! I hope we got that on video too. Okay?
Yep.
So thymine, all it does is hook up with adenine, and so guess what? We have just completed mRNA. It unzips from the DNA, and it leaves the nucleus.
Congratulations.
And it's swimming around inside of what?
The cytoplasm?
Cytoplasm, yes.
So is that all we're doing for the worksheet?
No, we got tons more.
We got
Okay.
tons
All right? Okay, yes.
Really? Did you do it? No.
more. No, we just.
Okay, can you hold off on that for a second? Think of that, because that's what I want to end at the end, okay, with what you just said. Okay? It's the difference between duplication and replication. We're in replication right now. Some people call this, I just translated the code from the DNA, and now this is going to unzip, and now it's going to float into the cytoplasm, and what is it looking for? What is it looking for? What's that?
Another copy of itself?
No. It's looking for something, or something is looking for it, and it's the most numerous thing in the cell. A ribosome. A ribosome is looking for that chain. Oh, there's a chain. The ribosome hooks into it, because what is inside the ribosome? rRNA. Very good. And so the rRNA is doing this. Oh, okay. This is what it says. We're going to make a replication. So it does this. It says, hello, come here. And now what comes in there? What comes into the thing? So, to give you an example, it's this right here. Okay, put your hand out like that. There's a ribosome, and here's the mRNA. So the ribosome, and it usually goes this direction from me. It'll go this direction from you. Okay? Okay, so now what happens is this. There's a ribosome, and it reads this. It reads this 3-base sequence, and then it says this. Come in here, tRNA, and it hooks up with it. Okay? And then it reads it, and then it takes the amino acid, which is the head of it, and it hooks on. It stays there, the amino acid. Then it takes another one. Oh, this one won't go to this one because it's different. So it's got to get a different code for there. Okay? So that's what it's doing. Okay? That is called what RNA? The tRNA. Very good. So now the tRNA is going to be involved with what's called transcription. Transcription. So now we're in transcription. So what we're doing here is this. The messenger RNA read the code, and it's translating the DNA, and now the tRNA, with the help of the ribosome, is putting it down into our language. Because right now this means nothing to the ribosome. But once it hooks up with there, oh, they've decoded it, now it's in a language. They've translated it. So now we got to get the next thing, which is called the tRNA. So what's the tRNA code to this? What goes with guanine?
Cytosine.
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 Adenine. Guanine. Thymine? Wait. Uracil? Oh,
why
yeah.
is it uracil? We're still in RNA. You can't have T if you're in RNA. So what's the next one?
Adenine. Uracil. Cytosine. Adenine. Uracil. Uracil.
Now you might be saying, Curvis, why do you have a gap there? Why do you have a gap there? There's a reason. There's a reason for the gap. So, we've got mRNA reading the code, coming out of the nucleus. It hooks into the ribosome. The ribosome is like a typewriter. It reads it, and it says, hey, it says, tRNA, come in here. That is called the anticodon. So, folks, this is the anticodon. It's also the tRNA. tRNA and anticodon are talking about the same thing. They're the same thing. Okay? Now, at the end of this tRNA, there's a head there. Because remember, the tRNA is this kind of a shape. Remember that? Remember that T shape? And at the end of it, it has a head. This head is the amino acid. This is called the amino acid. The amino acid is, get your table, get your genetic code table. The amino acid is this right here. It's what this is. So, that wheel is only from the mRNA. This is just what was brought in there from the tRNA. tRNA is not part of that wheel. Does that make sense? Okay? All right. So now we're coming on. We're going to come here and we're going to translate. We're going to translate this into amino acids. So on your paper, what is GGU? Glycine. So GLY. Okay? CCC. Proline. So PRO. AUA. Isoleucine.
That's hard to say.
And what is GUA? What's that? Is it leucine? Valine? And AAA?
Lysine.
Okay? All right. Does everybody agree with those? Okay? Now, these things hook together. These things hook together. They bond together. And together, this whole thing is called a what?
A codon?
No. These are amino acids that come from the codons, but what do they give us?
DNA strand?
No, DNA is at the top. You want to try? Very good. It's a protein.
I was thinking that.
Does that make sense now? The amino acids are hooked together in like bubble gum together, and they form a protein. So this is a protein right here. This is a protein when all the amino acids hook together. Okay? All right. So let's refer to this. tRNA, also known as the anticodon.
Transcription.
Yeah, but what is it called? Amino acids. Hook the amino acids together, they give me a protein. This is mRNA. This is DNA. Okay? Now, you got the question here, or do you have your other question? Your first question.
Oh, no. Does it, is it a certain number of amino acids that make up a protein?
That is very good. Okay? Now, I haven't got to the point here. It just so happens with this one truly is not a protein. Does anybody know why this is not a true protein?
It doesn't start with AUG?
It didn't have any AUGs.
Oh,
Remember the start one? Look at the start one. It's methiodine.
Yeah.
Is that what it's called? Methiodine? Methiodine?
Methionine.
Okay.
These words.
MET. You must have MET to start a protein. If you don't, they dissolve. And they float back into the cytoplasm. So do you see anything with MET here?
No.
So really it would not stay together. It would not bind. It has to have a start code. That start code is MET. There is no MET. And when will it know it has enough protein, enough amino acids to make a protein? When? What's that? Right, because what is GUA?
The end.
It's the stop. So once it gets GUA, it'll stop. That's the size of the protein, anywhere from 5 to 20, and that protein has its own thing of going to muscle cells, nerve cells, or something else. Okay? Well, there's two of them. There's two stops. What are the two stops? UAG and UAA and UAG are the two stops. So those two things was what you would be looking for in the mRNA. Okay.
You said that they wouldn't hold together, so like they just wouldn't be a protein.
Yeah,
what would
all of
happen?
a sudden, after you, when I went through her hand like this, I went like this, okay?
Yeah.
Okay, it comes through. If it's not MET, it breaks into the three things and they float inside the cytoplasm.
So
They're waiting for the next
sequence?
tRNA.
Oh, okay.
And so that's why you have so many ribosomes inside of a nucleus. There's tons of them because They're ready to be created all the time. Now, this happens in seconds.
Recycling.
It happens in
Nice.
seconds. This is happening all over the cell all the time. That's what's so cool about this whole thing. Okay?
Could you say, like, to describe it in another way, it's recycling?
Oh, did you hear what she said? That's a good way to think. It's recycling. Comes through. If it's not MET, they break apart and it's recycling. Waiting to find MET. Oh, but if MET was before this, then it would hook onto that one and the amino acids would stay in line. Okay? Then the bonds would be there. But the bonds aren't strong enough if MET doesn't start it. Okay? Yes?
Is there like some disease that...
That's mutations, and wait for mutations. That's tomorrow.
Okay.
Mutations is when MET gets screwed up, or MET is kind of switched around, or the bases don't hook up, their mRNA doesn't hook up correctly, and that's the different things that you guys study. Tay-Sachs. That's color blindness. All those things are because the base sequences did not line up the way they were supposed to, and so we have abnormalities. Okay? Yeah.
Okay, so you use the wheel on the second line, right?
You only use the wheel for mRNA.
Okay.
The wheel is only here. The wheel gives you this right here. Now, wait. Let's go back to your original equation. Okay? So now, DNA. It unwound. It unzipped. Because it wants mRNA to hook up to it. So mRNA hook up to it, then what does mRNA do? Once it hooks up to DNA and it finds a sequence, what does mRNA do? It doesn't do this yet. It leaves a nucleus. So what's this strand going to do? Well, that's where polymerase 3 comes in. Polymerase 3 comes in and says, if this is the beta chain, it needs the alpha chain. So it'll zip back. So it zips back because of polymerase 3. After it formed the mRNA goes out, and the DNA is already zipping behind it. So it's like your suitcase. You go like this, unzip it, it's making mRNA, and you're coming with this zipper right behind it, putting the DNA back together again.
Oh.
Okay, so now, what is the DNA? What is the DNA alpha strand if this is your thing? Because the mRNA is gone. It left the nucleus, and the DNA strand is there, getting zipped back. So tell me what the alpha strain would be.
I don't understand the alpha thing.
Thymine shows up. Okay, what about this?
ATA. GTA. AAA.
Triple A.
And some of these are not codons. These are that continuous thing. I wrote them as codons only for our thinking, but they're a continuous thing. They don't become codons until mRNA comes in it. Then mRNA, once it separates, it groups them tightly together in groups of 3 because of tRNA. All right? But, so you've got this right here, this long chain. But now, hey, remember when I talked about the interons? See, some of this stuff is just, it's recycling paper. It's garbage. It's not even going to read it. And so then all of a sudden the DNA zips back together, and all of a sudden the DNA says, oh, we don't need that, so it drops it out. Okay? And so that's where interons come in, because it's not important for the chromosomes. Okay? All right, go ahead.
Where does like the thymine come in if it's like year or so before?
Thymine never leaves the nucleus. So when it unzips, thymine's hanging around there. The thymine never leaves the nucleus, okay? And it unzips and it hooks back together here. Thymine is there.
But if it's on like the 2 strands, then like would it have to make more thymine?
Sometimes it will make more thymine only when chromosomes get weaker, but then all of chromosomes are there. But the thymine is always, when the strand unzips, one side has adenine, the other side has a thymine, right? Because when they were hooked together, adenine, thymine. But then they unzipped, thymine's still there, okay? And so it's going to hook back there, unless it's duplication, when you copy the chromosomes. When the copy chromosome happens during mitosis or meiosis, new adenines, new thymines come in, so you have two complete exact copies of each other. That's really replication. Okay? Well, we're into heavy stuff here, and I know some of you are going, oh, it's thick. Okay? All right. So now look at your. Okay, real quick.
Okay, I don't understand the whole alpha beta thing.
A DNA has how many sides?
2.
It has 2 sides. Okay,
Yeah.
we call those 2 sides, because it's a double helix, we call those 2 sides the alpha strand and the beta strand.
Okay.
Now when they separate, then they form two strands of double for a while, called mRNA. But if it's during replication, where we're creating a new cell, then we'll have 4 strands of DNA.
Okay.
Okay, and then we'll have 4 sides.
So is the one right above, like the DNA sequence right above the mRNA, is that like only one side of the double helix?
Yes.
Oh,
and the other
okay.
side is doing the same thing.
Got it.
The other side is creating another mRNA, but it's creating the other copy of this, which is different sets of proteins.
Okay.
Okay, all right. Okay, so now take out your sheets. Take out your sheets, and let's try some things here. Okay, I'm going to take the very first set of letters. What are the very first set of letters that you have?
TAC.
TAC? Okay. I don't want to get tacky on you. But, okay, I wrote them this way.
Cheesy.
I don't like it this way, but in order to put you in a box form, this is the only way that I could do to make that, okay? Now, what did I call this?
Tac.
Now, I called it something. The DNA strand
Strand
A.
A.
So this is my alpha strand of DNA. So before it starts to replicate,
Dude, I got
what
strand
is
B
it
right then.
doing? It's doing what I'm expecting on Tuesday or Wednesday of next week.
Like how he counts
Okay,
it?
your DNA molecules. What is it doing? It's hooked into
becomes
beta DNA.
T.
So it's hooked together right now with its other counterpart. So tell me what the counterpart is.
ATG.
ATG. This is what you should have with this. This is your double helix right here.
Up, I got it right.
Then all of a sudden it separates. And so when it separates, DNA is stuck inside that nucleus. It can't leave, so it needs what to come along?
mRNA.
It needs a messenger to carry information outside the nucleus. That's where mRNA comes in, and that's called translation. It's translating the code. Okay, what are you telling me, DNA? Come on, tell me. Speak in English, okay? All right, so
AUG.
what's the mRNA to this?
AUG.
AUG. No thymine. Okay? Yeah.
Are you making...
I am filling in the first box is all I'm filling in.
Now that I know what the hell I'm doing.
This all makes sense
The
now.
mRNA hooks in.
Kind of.
It's the mRNA is not a strand yet.
Seems a lot easier when I sound like.
This is inside the
Well,
nucleus.
I kind of guessed,
This
but
is
my
inside
guess
the
was right,
nucleus.
so I'm pretty
This
happy.
is inside. They're
Proud of myself.
individual nucleotides. They just come together to form a strand because the DNA strand is there.
But if... So, thymine is still inside the DNA, why you need mRNA to come inside?
Because the DNA can't leave the nucleus. Otherwise, how would your cells get oxygen? DNA doesn't tell you that. The mRNA's got to tell the cell, oh, well, let that oxygen molecule come in through the cell membrane. The DNA can't leave the nucleus. So the messenger, that's why the messenger mRNA is so important and it's ongoing. It never stops. It never stops. But now, mRNA, what's it looking for?
Looking for TRN.
No. It's looking for a ribosome. Because what's the ribosome going to do? It's going to put it together with what? tRNA. tRNA is also called, not its codon, this is a codon. Here's your codon, AUG, codon. But it's going to hook onto it of what? An anticodon. That's tRNA. What's the anticodon to this?
TAC. C.
C, good.
Oh, sweet.
Hey, folks, do you see anything here?
Yeah.
You notice that? Yeah.
Oh,
Oh, I'm sorry. You are so right. It is
Oh, it
you.
should be.
no. Wait, I mean,
That
What?
is very good.
the A is. Oh yeah. The A, B, U.
A, U, U to A, G to C. Okay?
What?
Okay, so here we go.
What?
The last box that you have, is
Acid.
the amino acid. Notice I put it here because really it's hooked to the TNA. The amino acid is this head of a tRNA. And it gets released after the anticodon hooks into the codon. It gets released. Now, what is the amino acid? Where do you have to go to find the amino acid?
To the wheel.
You have to go to chart, but you have to go back to the mRNA. Yes. So AUG. What is it?
The star word.
Yes, it's MET. So there is your amino acid. It can start producing a protein right from the start. Okay? How we doing?
I think I got it.
Good job. Good job. Well, we'll know how you do it because that's a worksheet for tomorrow. Okay?
So in the tRNA, the U, the A translates to the U because it's outside the ribosome? Or
No,
nucleus, I mean?
Because it's RNA.
Oh.
RNA can only have uracil and can never have thymine.
So
Now,
only.
here's a hint. Some people do this. Some people change everything to A, T, G, C all throughout the whole thing. That's what my teacher in high school told us to do. Change everything to T, and then if it's above or below RNA, to change all the T's to U.
That doesn't make sense.
So no, what he would say is this: change this to ATG, but since it's under RNA, erase the T and change it to U. Anything that's under the RNA. See,
Okay.
I don't like to do that because it's two different things. Okay?
Okay, I get it now, yeah.
So anytime it's with the RNA, you have uracil.
So it's only going to be the two original strands
Right.
that have.
And now
Okay.
chemically there's a reason why, and I'm not going into the chemistry
Oh.
part. Okay? That'd be a little bit upper level. Yes, sir.
Okay.
I don't understand how you did the amino acid.
Amino acid
It's the wheel.
is the name
Yeah.
of the codon. Where's the codon? It is the mRNA. It's not the tRNA. Okay, so now, you guys want a challenge?
Yeah.
Okay, here we go. Put that worksheet away. Here we go.
Snap.
Here's the challenge. And your classmates aren't here to see it. But you're going to get a paper on this tomorrow. Here we go.
Oh boy.
Quickly. Here we go.
Am I in your way, boys?
What's the anticodon? No. No.
What's the board say?
Yes.
Oh, yeah.
Yeah, you got it.
It's what?
God, that's gonna be a big ass.
UUC is the anticodon. It's the tRNA. But in order to find the tRNA, what did you have to do first? You had to
That's the
find the
tRNA.
mRNA, which is this. What's the mRNA? AAG. Okay? So tell me, if I give you lysine, what was the original DNA strand? What was the original DNA strand? TTC. Very good. The original DNA was TTC. The mRNA AGG hooked into it. It left the nucleus.
Got it.
It hooked in with anticodon UUC, dropped off the amino acid. Then it formed lysine, and lysine is waiting to see if there's methionine in before it, and then it can be a part of a protein. Wow, that's intelligent. Okay, you ready? You ready for
Yep.
the tough? Okay, here we go. Anticodon. I want to know what the DNA sequence is. I want to know what the DNA sequence is.
Why'd you put away your chart? Open up your chart.
It's TTT.
Yeah.
Yes!
Yeah.
Yes! That's learning! Yes!
Wait, what did she say?
Score one for the
Something
boys
Mariners
back
can't do.
home.
Wait, what'd she
Everybody who
say?
plays them can. Okay? All right. This is the tRNA because it's called anticodon. That means the mRNA is AAA, and it hooked into the DNA, which was TTT.
Smarty
Now,
pants.
when you can get this, you are in the upper echelon of being able to do DNA sequencing.
I get it.
Because I know college students that can't do this.
I'm proud of
They
myself.
just can't
I
do this.
did
They just can't. They can't grasp this whole concept. Okay. So that's nice stuff here. Okay. Here you go. You ready?
Nope.
Okay. Here we go. What's the What is the amino acid? What's the amino acid?
ATA.
Amino acid.
ATA. No.
This is the DNA. It's got to be DNA.
Oh, okay.
How do you know it's DNA?
Because there's
It's got T in it.
Oh.
It should be tattooed right on your arm.
long.
You were waiting for that one, weren't you? Okay.
Find TAT.
No, it's not you. So what's UAU? No.
What?
Oh no, you are right. You are right. You are all right beforehand. It is AUA, which is AUA is what? I just said you're right. I was thinking.
It's iso something.
Okay.
Isoleucine.
I don't know how to say it.
What's that? How do you know it's DNA?
Because it has T.
As soon as you see T in it, you know it's DNA.
Woohoo!
No, you can't. You don't know what the amino acid is until you put the mRNA in it. So you got to change this to mRNA. So the mRNA would be AUA. Would be the mRNA. So now we look for this for the amino acid. I could have said what's the anticodon of that. And that's what I was thinking. The anticodon of that is this. Keep all the letters the same. Change the T to U. UAU. So the tRNA, the anticodon, is the DNA without T. Oh, I applaud you. I applaud you because I have one of the hardest worksheets. I worked for 2 hours on it yesterday. I'm not going to give it to you today. I'm going to give it to you tomorrow. Okay? But one of the hardest ones. But if you get this one, okay, and you're going to be able to work together on it, okay, you've got it down. All right? Okay. Is the bell going to ring, Charlie?
We've been videotaping for 37 minutes, so.
Okay, because I have 2 videos that I would like to show you here. And what I wanted is I wanted a video
Class 40
tomorrow.
minutes today?
This class is 40
Yeah, we have math right after this. Okay, but here's the thing.
I don't understand.
You have this worksheet is due tomorrow. Tell your friends that if they say, "Well, I didn't know how to do it," say, "It's online." And you guys are the professionals now, so if they need help on it, you can help them. But here's the thing: don't give them the answer. Don't give them the answer because this is not necessarily easy. Now it's easy for you. Don't give them the answer. Show them how it works. If you just give them the answer, then that's not going to be helpful to them. Okay? Have a good day. Yeah.
That was the best biology class ever.